So DNA, which is called deoxyribonucleic acid, kind of like carbohydrates, this name describes a little bit of the composition. So we'll get into that a little bit later. Before we learn anything about DNA, we're going to learn about the history involved in the discovery of DNA, okay? So there was a couple of scientists that did amazing work, and we have to give them credit for their discovery, so we're going to go through a couple, okay? And I have pictures of all of them for you. So this is Griffith and Avery. He's called Frederick Griffith, and the other one is Oswald Avery. I was going to say Gravy. Even though they didn't do the same thing, Griffith started an experiment, and then Avery kind of piggybacked and took the experiment to another level. What you have to know about both is that they discovered that there can be transformation of material, okay, from one organism or within cells of one organism to another, okay? They really don't know, or I would say Griffith or Avery at the time really don't know what genetic material is that's being transferred. Griffith actually thought that protein was what contained genetic material, and Avery later proved what? What contains genetic material? Good, DNA. What their experiment entailed. So you have to know the following. You have to know that Griffith and Avery discovered transformation, okay? You have to know that Griffith thought it was protein. You have to know that Avery thought it was DNA, or proved, I should say, even though he's still thinking because he doesn't really know. He just did an experiment, and they both experimented on mice, and they used strains of bacteria, one that was a live strain, one that was a non-live strain, and then they saw what would happen to the mouse if it obtained that strain. So I'm going to show you a video that'll kind of explain it better than I would. So Chargaff basically determined that the structures within DNA match up in concentration. And what that means is, and this is hard to understand because you don't know what the structure is, but he says adenine is a part of DNA's nitrogenous bases, and the amount of adenine is always going to equal the amount of thymine. Anybody remember these words? What does G stand for? No. Guanine equals cytosine. Okay? So Rosalind Franklin, what does this look like to you, by the way? No, when you see a picture like that, that's a picture of something.
Oh, it's like radiology.
Good. That's exactly what it is. She used radiology or X-ray diffraction to discover the shape of DNA, and she saw that it was kind of like an X-shaped, laddery looking thing, right? Sadly, she passed away, and then these guys actually ended up with the Nobel Prize, Watson and Crick, for making the first model of DNA. Hold on a minute. You have to know that Rosalind and Watson and Crick are the discoverers of the shape. You want to know that Rosalind used X-ray diffraction, and Watson and Crick built the first DNA model, which is right here, a picture of them and their model, and actually ended up with the Nobel Prize, okay? So DNA, deoxyribonucleic acid, is a macromolecule. What does it mean to be a macromolecule? Remember we learned 4 of those already? A big molecule. It carries genetic instructions that give information for the cell to grow, develop, function, okay, and reproduce. But in addition, something you might have not known, because most people know that DNA carries genetic information and it's involved in reproduction and it's how you get your traits and all that. But DNA also makes very important things in your body. What do we know about amino acids? Good. Amino acids are the building blocks of proteins. DNA makes protein. Good job.